Hello everyone! Today we are talking about cell division. We're talking about mitosis and meiosis, which is just how our cells make more cells. Because, obviously things grow, so we need cells for that. We can't just have one cell for the rest of our lives because that won't get us very far. So in the last video in this playlist, I didn't actually make the video. It's in this playlist linked up in the corner. And you should have learned all about the cell cycle. So if you haven't watched that video, I would recommend you watch it now because we're gonna say a couple things that you will need to know. And actually I'm gonna turn this page. So we're just focusing on one page right now. And let's talk about mitosis. Okay, so in that other video that I have linked in that playlist, we learned about the cell cycle. So how cells prepare to divide. When cells are preparing to divide, they will make more organelles, they're gonna make more DNA, because if they don't make more DNA and they divide, they won't have enough DNA for both cells. So we're gonna assume we've already gone through these stages, and that's gonna prepare us for doing something called mitosis. So mitosis is just the basic duplication of a cell. If you're a child and you're growing, you're gonna want more cells, like more blood cells, right? So one cell is going to divide and create two identical cells. So this mitosis is the division into two identical daughter cells. And daughter is just like what you say to refer to two cells that come from one parent cell. And when we say identical, we're really referring to the genetic information of a cell here. So that last video talked about the cell cycle. We replicated our DNA and organelles, and that process is interphase. So here we have our cell, and I'm gonna draw the nucleus, and the nucleus has all our DNA in it, right? And I'm just going to draw like a bunch of scribbles because DNA, we often see it in that chromosome structure, which I'll also link to a video about DNA terminology, which is important for understanding the terms that we use here, which you should definitely watch. So our DNA is packaged in this stuff called chromatin, and we'll often see it condensed as chromosomes, but that's not how it usually is in the cell. The vast majority of the time that the cell is existing, its DNA is arranged in chromatin that's all like jumbled up. It only condenses during cell division, which we're going to see here. So in interphase, it's not like that yet. And I'm going to draw these circles. So I'm just writing a C in each of them. So what these are supposed to be, so when I say C, what I really mean is, I just don't want to draw it every time because it's just a little bit tedious. These are called centrosomes. So a centrosome is an organelle that contains two centrioles, which kind of look like this. So this is a centrosome. These are centrioles. And they're important for cell division, for moving around chromosomes and chromatids, and we'll talk about that when we get there. You'll see what I mean. Okay, but right now they're not doing anything, they're just kind of sitting there. But this is interphase. So as a brief synopsis from the video that hopefully you have watched, we have the G1 stage, which is for growth. We're making more organelles. If we only have if we have 5 mitochondria in one cell and we want to make 2 cells, we'll make 5 more. So each daughter cell has 5, making a total of 10 before we divide. And then we have S phase, where the DNA duplicates. And the G2 stage, which is also growth. And then we move on to the M phase, which is mitosis. And so mitosis is composed of many steps. So you may hear the acronym PMAT. And that's what we're about to get to with our next step. So our first one, the P, is for prophase, which I think is nice because the first step having pro as a prefix is kind of nice because we know pro, like prototype, that's like early stages, beginning, before, So prophase, our cell. So we move forward in this little cycle. So here we have one of our centrosomes, and it's going to be releasing these things called spindle fibers, and we're going to have those on either side of our cell. And you'll see why in our next stage. But another thing that happens is our nucleus changes a little. So our nucleus here has this strong border. Now I'm drawing this dotted border to represent this nuclear envelope disintegrating. And also our chromosomes are going to form from the chromatin. So the chromatin is going to condense into chromosomes. I'm going to draw 4 chromosomes. Most, things have more than 4 chromosomes. Humans have 46. That's 23 from each parent. So a lot more than 4, but I don't want to draw 46 chromosomes in every single step. So we're just going to draw 4 here. So what are the things that have happened? Okay, our nuclear envelope is disintegrating. Our chromatin is turning into chromosomes. And finally, the spindle fibers are extending from our centrosomes. Okay, so what is our next step? What happens next? And why are these spindle fibers extending? Okay, now we're going to the M in PMAT, metaphase. And this one also kind of has a nice name, because M, I feel like, kind of stands for middle. So what's going to happen here is our chromosomes are going to all line up in the middle. So you have your centrosome, and it's going to be taking these spindle fibers to pull the chromosomes into the middle. And there's some spindle fibers that are reaching but aren't connected to anything. And so now they're all lined up in the center. And why is that a thing? Well, we will find out in our next step. But right now, what this does is our chromosomes are lined up in the middle. And that's done by the spindle fibers. So just to reiterate, the spindle fibers come from the centrosome, which is this organelle with centrioles. I know, it's a lot. And we can do some color coding. And if you just want to color it in, make it look a little bit more aesthetic, you are welcome to. Let's say we're representing the centrioles here. All right, what is our next step? Our next step is anaphase. So before, we had our chromosomes lined up in the middle, and now these spindle fibers, each spindle fiber is going to take 1 chromatid. So you watched the DNA terminology video, right? You know what a chromatid is? It's 1/2 of this chromosome. I've drawn the circle a little larger than I needed to, but it's fine. We have plenty of space on this page. Okay, so each spindle fiber, you can draw them all reaching out. Each spindle fiber is going to take 1. So 1/2 of this chromosome, basically. So what's happening here is our spindle fibers are pulling the sister chromatids, which are identical. Chromatids to opposite poles. So opposite sides of the cell. As you can see, the identical chromatids are going to the opposite poles. Okay, next step, we're getting there. So now we have all of the genetic information going to this side and this side. So we're preparing to split off into 2 cells. So we can draw our little cell being kind of the shape is starting to form of 2 separate cells. And basically the whole point of this next step, which is telophase, is the reverse of what happened in prophase. So in prophase, our nuclear envelope disintegrated. Now our nuclear envelope is going to reform. And also our chromatin condensed into chromosomes. Now it's going to decondense back into chromatin. in this spirally structure. And we still have one centrosome on each side. So our nuclear envelope reforms. And the chromatin decondenses. Okay, cool. So that's telophase. And now it looks like we're basically ready to be two individual cells, our daughter cells. So the last step is just splitting off, kind of closing this gap to become two independent cells. And that's called cytokinesis. Which kind of makes sense because this is the cytosol, right? This cytosol fluid. And now we're drawing a barrier in between to separate the cytosol. And so now we have two independent cells. That one is really small. I don't know why I drew that one so small. Don't do that. Make them look the same. And we have our decondensed chromatin and one centromere each. I guess you can draw like these, like pulling back into the centromere if you want. And now they're fully withdrawn. So you have your two daughter cells. Hooray! And there's a couple of different ways that you can see cytokinesis happening. In animal cells, it'll look a little different from plant cells. So we can draw that because that's all that we have left for this little spread. So as we drew with telophase, we drew these cells kind of like glomming out, like having that pressure point and then separating into two cells. That's what's going to be called our cleavage furrow. When we learned this, we thought it was very silly because it kind of looks good, but and that is great for animal cells. But plant cells, they're all rigid. They have that cell wall, you know, very strong walls made of cellulose. Very, very nice for your structural support, but it makes it hard to, you know, separate like that. So what they do instead, they deliver the materials to make a new cell wall into the middle of the cell, and that forms the new cell wall, and this new forming wall is called the cell plate. So yeah, that's how we get two new plant cells, and that's a little different from animal cells. This whole thing that we've drawn here is very much what animal cells would probably look like. So let's review. What have we learned? So interphase has the G1, S, and G2 phase. Both of the G phases are for growth, you know, like organelles, and S phase is for DNA duplication. Reminder to, if you haven't seen the video on this, you should check it out. In prophase, our nuclear envelope disintegrates because we got to be moving our pieces of DNA around, so if it's all locked in there, that's not going to work too well. And our chromatin condenses into chromosomes. We also have these spindle fibers starting to extend from the centrosomes, which are over here. In metaphase, the chromosomes are lined up in the middle, which is perfect because there's an M for metaphase, and they are pulled by the spindle fibers. And then what happens in anaphase is those spindle fibers grab each chromatid from the chromosome and pull it to one side of the cell so we can divide in telophase. So they are identicals. That's important because remember, we're making two daughter cells that are the same. We're just duplicating our cells to opposite poles. And then in telophase, our nuclear envelope reforms and the chromatin decondenses. So telophase really is just the opposite of prophase. And then cytokinesis, we split into two daughter cells, and you can do that either by like a cleavage furrow or your cell plate, depending on if you're an animal or a plant cell. And don't ask me about fungi, because I don't know. So this is mitosis. That's like half of the cell division that we're talking about. What happens to the other half? Well, that is meiosis. So we're still talking about cell division. Cell division. So what is meiosis in comparison to mitosis? So mitosis, remember, we were making two identical daughter cells. But in meiosis, we actually don't want identical cells. And you may be asking, why wouldn't you want your cells to be the same? Like, what kind would they be then? Meiosis is done for reproduction. So if you have a cookbook, you have two cookbooks, and they're really pretty good cookbooks, but you want one cookbook that's like the best of them all. And so what you want to do is you want to combine a bunch of recipes from each cookbook and see which combination makes the best cookbook. So let's say you take a few pages from one and a few pages from another. You'll still have the same amount of pages in your breakfast, lunch, dinner sections, but it'll be a mix of whose pages you'll be taking. So you'll make a bunch of new cookbooks that have different combinations of different pages from the other previous cookbooks, and based off of how well they perform, how many people buy them, you can see which ones are the best. And that's kind of similar to how we do reproduction as like organisms. So when two organisms breed, their offspring will have some alleles from one parent and some alleles from another. And the ones with the best combinations will have the best traits, so they'll survive the most and go on to reproduce. If an animal were to just reproduce by making a clone of itself, the types of animals we would see would not be very diverse. So this kind of gives life the opportunity to have a vast array of different traits, and the ones that are the strongest will survive and go on to produce new offspring, and then you can see different species developing, and that's evolution. So how do we do that? How do we maintain that genetic diversity? Well, the overall process doesn't differ too much from mitosis in terms of, we're still going to be talking about prophase, metaphase, anaphase, telophase, and cytokinesis, but it is going to look a little different. So let's start out by drawing prophase. Meiosis actually has prophase 1 and 2. So this is prophase 1, of course. It's not confusing like photosynthesis, where the first photosystem is actually the second. And we kind of got a similar thing going on here. We have our centromeres on opposite sides, reaching out. And we have our nuclear envelope disintegrating. Okay, that looks good. And then we're going to see something a little weird happening. So I'm going to still be drawing 4 chromosomes, but now what you want to know is this chromosome and this chromosome are called homologous. So what that means is they encode for the same genes, but they have different alleles. So what that means is both of them might be encoding for something like your skin color, but one of them will have maybe a darker skin tone and one will have a lighter one. So these chromosomes are basically matching. Humans, we have 23. Each chromosome is different. It encodes for different things. And so if you're looking at your parents' cells, your first chromosome, chromosome number 1, those will be the same, but they'll have different information on them, but they code for the same traits. Like in our recipe book analogy, they're both coding for breakfast, but maybe one of them will have pancakes and one of them will have waffles. Okay, so something weird is happening in prophase 1, and we're gonna call this crossing over. So I'm just gonna draw like a line to say like this is happening right now. It's not an arrow because we're not like going on to the next step, because this happens during prophase 1. Okay, so this is crossing over. Oh, I forgot to be writing in my marker. It's okay, we'll highlight this one instead. Okay, crossing over. So this is still part of prophase 1. We're gonna look at one pair of homologous chromosomes right now. So say there's one that looks like this, it's a lighter color, and there's one that looks like this, it's a darker color. So what's happening here is they're kind of overlapping. And what they're gonna do is they're gonna swap some genetic information. If we're talking about our recipe book analogy, this is pages like 10 through 15, and we're gonna swap the contents of each recipe book. So this is book 1. Now pages 10 through 15 are from book 2. And likewise, in book 2, pages 10 through 15 will be from book 1. So we swapped. And this is a really good way to see, okay, so if this is our breakfast chapter, maybe swapping just some of the pages will improve our book. And also, in case you need to know this vocabulary, one set of 2 homologous chromosomes are called a tetrad because there are 4 chromatids. Here's a video on crossing over so you can actually see it. So now that we know that's what happened in prophase 1, let's go to metaphase 1. So we have our cell once again, and we can draw our crossed over chromosomes. So this is our book 2 with a couple pages from book 1, and this is our book 1 with a couple pages from book 2, basically. And then maybe a similar thing happened over here. Maybe a couple different pages got transmitted. Okay, so we still have our 4 chromosomes, but they've done some crossing over, so that's pretty cool. So now they're lined up in the middle, but they're lined up by pairs. So before we had like a chromosome 1, 2, 3, 4, now it's 1, 2, 3, 4 because they're in their homologous pairs. And so the centromere is going to come over and grab one of each And as you can see, if you can imagine what anaphase is going to look like, it's going to pull these chromosomes and they're not going to match on either side. But that's what we want. We want genetic diversity. Each gamete that we form as a result of meiosis is going to be different, which results in a whole host of different offspring you can have. So metaphase 1 happens, and then we go to anaphase 1. And I'm not writing down all the things that happen that are the same with mitosis because, like, yes, the nuclear envelope's disintegrating, we're lining up in the middle, but that's the same principle. The things that I want to write down are the things that are changing. Okay, so yeah, the chromosomes get pulled to either side. This is anaphase 1. This concept is the same as it was before. It's kind of hard to see because this is a little bit small, but we're gonna have our same chromosomes just being dragged to different parts. Anaphase 1 happens, we drag the chromosomes to one side of the cell. And so we can see what telophase is gonna look like. If you feel like you really understand like this process, I guess you can skip these stages, telophase, cytokinesis. But I think it's nice to remind yourself of the overall like idea of what each step is, because that's the same in mitosis, and what it's going to look like with our different kind of chromosome situation happening here. So just like before, our nuclear envelope is reforming. Gets a little hard to keep all the details. So we're preparing to divide. We have the very different genetic information on both sides for when we divide. So we go through cytokinesis to have two cells that are distinct. And each of them, remember, if this is book 1, book 2, if this half of the parent DNA is book 1, and this half of the parent DNA is book 2, then, and like one chromosome maybe represents the breakfast section, this would be the lunch section. Each daughter cell has a chromosome for each part of your recipe book still. But now the pages are going to be a little bit different because they come from two different sources. But if you remember our chromosome structure, each one of these chromatids that make up a chromosome are identical. Now they're not identical because one of them has some of the pages swapped out, but this is still mostly composed of book 2. We only want one copy of each book, so we're going to divide again. And I'm not going to draw all the steps. We're not going to have prophase, metaphase, anaphase, telophase again. I'm just going to draw this going into two daughter cells. So assume that we have the whole PMAT happening, and now we have two daughter cells. For visual purposes, here's another one of those videos. It's of meiosis 1 and 2, and you can see us forming those haploid daughter cells. So this is just based off of this one cell here. You can see that there is 2 full chromosomes made out of 4 total chromatids. So what's going to happen is we're going to separate one chromatid from each chromosome. So here we can see what I did is I took this piece, and that went onto this cell, and then this piece and this piece went onto this cell. So you can see that they're a little bit different. This one contains some mixed up DNA, and in reality, crossing over can occur more than just once. So what will probably happen is both of these will have mixed up DNA, but one will be mixed up in a different way from another. So we're gonna have two daughter cells that are completely different. And these daughter cells are completely different from the ones that would arise from here. We can just draw that to make sure we know each cell that we start with ends up with 4 different cells. This is really small. Maybe I shouldn't have drawn it so small, but we're not looking too closely at this one. So we end up with 4 total gametes, and they're all completely different from each other. So the idea is, if you have the gamete of one individual and it mates with another individual, and when they combine, they'll have 2 sets of DNA from each parent. So once again, you'll have 2 cookbooks that you can swap recipes or genes out of. And that's the whole point of meiosis. We make gametes, but we only end up with 1 copy of each gene so that we can combine with another gamete from like if this is from the mother, it'll come from the father, to have 2 sets, so we can have genetic variation. So by using only half the DNA, we allow for genetic diversity, because we can combine with other individuals that also only have half to add up to one whole. And a couple important notes is prophase 1 has crossing over, but in prophase 2 we don't have that, because if we start with a cell like this, who are we going to cross over with? If these are our recipe books, this is the breakfast section of book 1 and this is the breakfast section of book 2, there's no other breakfast section. So who are we supposed to cross over with? I can't cross over with lunch, because then I'm going to have lunch recipes in the middle of my breakfast section. That's not going to work. So crossing over only occurs in prophase 1, not prophase 2. Also, you may be wondering, when we line up for metaphase 1 and we get pulled into separate cells in anaphase 1, how do we know for sure that we're going to have one breakfast section of each and not like 2 lunches and 2 breakfasts going into each cell? Because that's not the kind of recipe book we're trying to make. Well, fortunately, there's a law of segregation which ensures that our 2 different types of chromosomes segregate into the proper halves. So matching chromosomes, these homologous chromosomes that both encode for breakfast, each one is going to go into one of the daughter cells. So each daughter cell gets 1 copy of each homologous chromosome. So as you can see, we are getting 1 of each breakfast section, 1 of each lunch section, and when we go and do cytokinesis and we move on to metaphase, anaphase 2, we will still get 1 lunch section each. It'll just be a single copy. So instead of chromosome, that would be a chromatid. All right, so that is meiosis. We are making gametes so we can have new individuals. So this only happens in sex cells. You're only making gametes in your germ cells. So this would be like where females produce egg and males produce sperm. You are making cells like this so that you can reproduce. When 2 gametes from different parents combine, it'll form a zygote with 1 set of each parent's DNA. So when you start, you have a cell that's called diploid, meaning it has 2 sets of chromosomes for each trait. So both breakfast, through this first stage of meiosis. Now each section is only from 1 recipe book. So now you're haploid. And I'm using N here to represent the amount of each chromosome. So here we have 2N because there's 2 different types of books, and now there's only 1 type of book for each section. So these gametes are also haploid. They still only have 1 breakfast section, but when they combine with another gamete from another parent, they will form a zygote with 2 sets of each, making it diploid again. So that's why we only have half. And that's how it increases genetic diversity. So yeah, remember, crossing over is only a prophase 1 thing, it's not a prophase 2 thing. And the law of segregation states that each daughter cell will get 1 copy of each homologous chromosome, or chromatid. All right, and if you don't remember these terms, homologous describes 2 chromosomes that contain the same genes but different alleles. So they are both the breakfast section, but they might have different recipes. Same genes, different alleles. All right, so yes, that is mitosis and meiosis. That is the basics of cell division. Hopefully my little recipe book analogy made sense. I've never heard that one be used before, but I think it's a good way to explain chromosomes and genes and alleles and how they're different and how that is important. Make sure you check out that playlist with the other bio videos. We will continue the series, but unfortunately without me. The rest of the videos are actually done by Miss G. You can see them in the playlist, but cell division is actually where I leave off. But I hope you enjoyed. Leave a comment if you have any questions. And yeah, thanks for learning with me.